Sound check.
All right. Check.
Good. Check.
We're good?
Okay. Let me check.
Aloha. My name is Brian Bowen, and I'm a researcher at Hawaii Institute of Marine Biology. The University of Hawaii Sea Grant College Program, otherwise known as Hawai'i Sea Grant, is based at the University of Hawaii at Manoa in Honolulu, Hawaii. Hawai'i Sea Grant supports an innovative program of research, education, and extension services directed to the improved understanding and stewardship of coastal and marine resources of the state, region, and nation. Hawai'i Sea Grant, podcast number 1, Fish Flow.
Aloha and welcome to our 321 podcast. My name is Mahealani.
And this is Rachel.
Brian here.
And I'm Michael.
And we are part of Hawai'i Sea Grant and the University of Hawaii located in Honolulu, Hawaii.
This podcast features research supported by Hawai'i Sea Grant. Each episode will highlight a new project and the people that make that project possible.
We will ask these researchers for 3 words that describe their research. Give them 2 minutes to explain how their research benefits communities here in Hawaii, and end by asking them to provide one thing they know about each other than their research. So, 3 words, 2 minutes, and 1 thing. It's a 321 podcast.
Hey, Mahealani.
Yes, Rachel?
Have you ever seen baby fishes when you snorkel?
Baby fishes? Like itty-bitty fishes?
Yeah. I think I've seen a lot of juvenile fishes, but those are already big.
It may be hard to see baby fishes, or what researchers call larval stages of fishes, since they're tiny and clear.
This helps them blend into the environment and avoid predators.
So, you're fish guys, right?
That's one way to say it.
That's us.
What are 3 words that describe your research?
Fish, connectivity, dinner.
In other words, spawning to table.
That's good. All right. So when you talk about spawning to table and you're talking about dinner, I guess you guys are talking about fish species that people like to eat.
Yeah, definitely. So one of the species that we're looking at is manini.
We're also looking at kolei.
And finally, we're focusing on the yellow-lined goatfish, or known in Hawaii as beke'a.
In this podcast, we're learning about fish and how they flow. And here to share their research are Dr. Brian Bowen and his graduate student, Mykle Hoban.
Welcome and aloha.
Thank you.
Thank you.
Before we ask you how your research benefits communities here in Hawaii, let's talk about a few scientific words. So, 3 words that you use a lot in your research are spawning, settlement, and connectivity. Can you explain what these words mean?
Spawning refers to fish reproducing. And unlike land animals, fish reproduce out into the water. We call this broadcast spawning. And when this happens, the males and the females release eggs and sperm up into the water, where they meet and the eggs are then fertilized. After a short period of time, the eggs hatch into larval fish, and they also spend a significant period of time in the water.
Settlement refers to the process where they transform from larvae into little fish that actually look like the fish. And they drop out of the water and they settle down into a reef.
So when you mean settle, is it like they're making a home?
Well, it's not settling like a coral, which will find a place and stay there for a lifetime. But they'll settle onto a reef and establish a home territory of maybe an acre.
And finally, connectivity draws the connection between spawning and settlement. Because the fish spawn into the water column, and because the larvae spend a certain amount of time in the water column, they often settle in a different place from where they spawn. What we're trying to do is figure out where fish come from and where they go.
So in 2 minutes or less, can you tell us how your research benefits the communities here in Hawaii?
Well, we know that some places are really good for producing fish, and some places the fish accumulate. If you think about it for a bit, where do you want to go fishing? Do you want to go fishing where they're spawning and take out those adults that are producing the next generation? Or do you want to go fishing where they accumulate and there's a lot of them there that you can take?
So unraveling these connections allows us to draw a map of where these different locations are, where they come from, where they go. And ultimately that allows us to better conserve these fishes so that we can keep bringing them home and putting them on our dinner table.
So one of the products of your research at the end, if everything goes smoothly, because research always goes smoothly, right?
Always.
So
As smooth as sandpaper.
Yes. So when Mykle graduates, he'll have a map of where to fish and where not to fish.
That's a really good way of putting it.
That may or may not be an area that local fishermen know where to go.
Yeah,
Right?
that's true.
So, because fishermen, they like to have their go-to places, right? So what if that go-to place was actually an area where they're spawning and maybe they're caught before they spawn? That would be a problem.
Yeah, and that's where podcasts like this are important because we can get that information out to people and hopefully it doesn't come as a threat to their livelihood, but rather it shows we're looking out for you and how you can continue to do the things that you're doing into the future.
Eat the fish you like to eat.
Precisely, and have more fish to eat.
Let's recap. When asked to provide 3 words that describe your research, you said spawning to table. You provided definitions for 3 scientific words that are commonly used in your research. These words were spawning, settlement, and connectivity. We learned about your research and how your research will help the communities here in Hawaii.
That's right.
That's what we're doing.
So let's get technical. How do you do your research?
We primarily use DNA to do our research. And the reason for that is because these larval fishes, as we said before, are very small and they're clear, which means they're very difficult to find in the water column and they're virtually impossible to track. In a perfect world, we would attach a satellite tag to all of the millions of baby fishes and track them everywhere they went, and then we would draw a perfect picture. In reality, because that's virtually impossible to do, we use DNA. And the way that we use DNA is we look at the DNA of the settlers, the young juveniles as they first arrive on the reef, and we look at the DNA of the adults, and by comparing how related those are to one another, and by comparing that across different areas around the island, we can draw that picture of connectivity. Because if the more closely related adults are to juveniles, that means it's more likely that the adults will be the parents and the juveniles are the offspring of those parents. So we can draw those connectivity lines and develop that map.
This is a version of DNA fingerprinting, the same technology that's used to establish the guilt or innocence of a suspect in a murder trial.
So, you catch the fish. Do you have to use the whole fish?
No, so DNA is in every part of the animal. And so with fish, all we have to do is cut off a little corner of one of their fins, which will grow back or it does not affect their ability to swim around in the environment. And so we take that little piece of fin and we chemically extract the DNA from that.
How do you catch the fish? They're awfully fast swimmers, aren't they?
We had a great deal of difficulty finding the Becca until we listened to the local fishermen.
And the local fishermen, their knowledge is deep and comes from a long tradition. And so we have a lot to learn from them. And so we try to reach out to them and learn from them wherever we can.
Well, that's a wrap. But before we go, we want to ask you, Brian, what is one thing you know about Mykle other than his research?
Tats.
Tats? Yeah.
This guy has some fantastic tattoos.
How many tattoos do you have?
I don't count, but it's certainly, I'd say it's more than 20 and fewer than 100.
They're all over you. Wow.
All right. And Mykle, what's the one thing you know about Brian other than his research?
know that he's extremely fond of the Grateful Dead.
Oh, yeah?
Oh yeah, they're touring this summer.
Are you going to go?
I'm gonna try. They're playing on the West Coast.
I thought Jerry Garcia was dead.
Well, that's the name of the band.
All right, well, stay tuned for our next podcast, and mahalo for listening. Aloha.
Aloha.
This Hawai'i Sea Grant 321 podcast featured 3 songs: Happiness by bensound.com, Ukulele by bensound.com, and Cute by bensound.com. Find it on iTunes or bensound.com.